This is a production of Cornell University.
Thanks, Jim. I appreciate it. I wish I was dressed like Jim right about now, to be honest with you. So I can offer pretty much unambiguous proof for global warming. When I was an undergraduate here back in the early 80s, I don't think in the 4 years I was here it ever got this hot in the entire 4 years I was here. So it is warm out there. I guess I bought it from North Carolina. So today I'm going to tell you about cytokinin signaling. Mostly, unfortunately, no ethylene today, Jim, but we're going to talk mostly about cytokinin signaling. Where was I? Yeah, so this, cytokinins were discovered, of course, as most of you probably know here, from the pioneering work of Folke Skoog and Carlos Miller working in Folke's lab. And they discovered the existence of cytokinin in the 1950s. And their initial discovery was that cytokinins, in concert with, of course, this other hormone, auxin, promoted cell division or cytokinesis, hence the name. And they made two seminal observations about cytokinin. One is that cytokinin in concert with auxin could promote cell division, but also that the relative ratio of these two hormones determine the differentiation of organogenesis from undifferentiated culture. So this is tobacco cultured cells, and you can see at relatively high concentrations of cytokinin, shoot-like organs form, that eventually shoots would form. At relatively high auxin concentration, roots would form, and then at intermediate or high concentrations of both, you'd get this undifferentiated growth. So cytokinins were discovered in the 1950s. After that, there was quite a bit of work going now. All right, so since they were discovered, quite a few roles were elucidated for cytokinin. Some of it was from exogenous application, but a lot of it was derived from analysis of our favorite model system, Arabidopsis. Much of this was the result of loss of function mutations that made plants insensitive to cytokinin. And like pretty much any of the phytohormones, somebody could show a slide just like this. If you worked for ABA, or if you worked for auxin, if you worked for GA, you're going to show a slide just like this, which basically highlights the pleiotropy or the pleiotropic nature of hormones, that they basically do or affect almost every developmental and growth process in plants, and cytokinin is no exception. And so they do things like, for example, promote cell division in the shoot apical meristem. They actually inhibit cell division down in the root apical meristem. They, as Shushen knows, they inhibit leaf senescence. They regulate oxalate bud growth, and so on and so forth. So they do many, many different things. And so the question that we addressed quite some time ago is, how is it that plants perceive cytokinin and transduce the signal? And then an offshoot of that question is, how does one simple signaling molecule do so many different things? So in different tissue, it has completely different roles. Okay, so to get at this question of how plants perceive cytokinin, a couple years ago, actually probably a couple decades ago now, two independent lines of investigation led to really the elucidation of the cytokinin signaling pathway. One was done by a very talented researcher, Tatsuo Kakimoto in Japan, and he was screening for things that allowed Arabidopsis, in this case, to form shoots in the absence of exogenous cytokinin. So normally in a wild type plant shown on the left, if you don't give any, as I told you before, if you don't give any cytokinin and you give auxin, you get lots of root growth. And then if you give auxin plus cytokinin, you induce shoot formation in culture. He found a line that in the absence of exogenous cytokinin forms shoots. He called it CKI1 for cytokinin insensitive. And as you'll see in a moment, it was the result of the overexpression of a gene called a histidine kinase. Now, it turns out that the gene he's overexpressing here, CKI1, almost certainly does not play a role in cytokinin signaling, but paralogs of CKI1 do. In parallel, we were trying to find genes that were rapidly and specifically induced by cytokinin, and we used a technique that probably nobody in this audience knows, called differential display, which doesn't matter what it is, but it was a way to identify genes that were induced in response to a particular hormone. Now, back when we first started working on cytokinin, the cytokinin responsive genes were induced with treatment of cytokinin in about 6 or 7 hours, a long-term treatment. We wanted to find a primary response gene, the first genes that are targeted after you give exogenous cytokinin. And we identified a gene that is now called AR5 that was rapidly, and at least rapidly, the rapidity was specific for cytokinin. And for the young undergraduates and their graduate students, that's called the northern blot. Nobody ever does northern blots anymore. In fact, everything I did as a graduate student postdoc isn't done anymore. But anyway, so we found this gene called AR5. And these two lines of investigations converged in the same kind of signaling pathway, so-called two-component signaling pathway. These are ubiquitous in bacterial cells. So you know that animals and other eukaryotes, plants, have things like serine threonine kinases, and animals anyway, tyrosine kinases. In general, bacteria don't have such elements. They have these two component elements to perceive and sense the world around them. And as the name implies, they're comprised of two elements, a receptor called the sensor histidine kinase, and an output signaling element called the response regulator. And so the basic idea is that the input domain binds some signal, whatever that might be, low phosphate or whatever. In bacteria, it regulates the activity of a histidine kinase domain, which autophosphorylates in a histidine, and that very same histidine is then transferred to the aspartic acid residue on the receiver domain of a response regulator, which oftentimes acts as a transcription factor to regulate gene expression. So a very simple signaling system where you have a receptor directly regulating the activity of a transcription factor. Tatsuo's work, where I showed you CKI1, Tatsuo's work identified a sensor histidine kinase, and our work identified a response regulator. So that immediately suggested that the way that cytokinin is perceived in plant cells is by a two-component signaling pathway. Now, if you look actually in eukaryotic cells in general don't have a simple version of the two-component pathway. They have a slightly more complicated version called the phosphorelay, which is very similar, except you add on a pair of extra phosphotransfers. It still alternates between a histidine and an aspartic acid. And in general, there's a small phosphoshuttle protein called a HPT, or histidine phosphotransfer protein. And this is the way that cytokinin signaling works. There's a receptor that's a hybrid sensor kinase that transfers ultimately a phosphate to a AHP, which then results in the phosphorylation of an AR protein. Okay, so this was sort of immediately apparent from those two lines of investigation. We focused in the beginning on these AARs. Turns out that these ARs, these response regulators, are a fairly large gene family in Arabidopsis and other plants. They fall into two very clear clades, two very distinct clades, and we very creatively call them the type A and type B clades. They are different both in terms of their sequence similarity, they're different in terms of their domain structure. The type Bs consist of both a receiver domain and a well-defined output domain that acts as a DNA binding element. So this domain binds DNA and has an activation domain. So this is a DNA binding transcription factor. In contrast, the type A's really are just comprised of the receiver domain with in general, very short C-terminal domains. And then finally, the other difference is that the type A's are strongly induced by cytokinin. I showed you the northern blot before. The particular one I showed you was at AR5, but most of them are induced, as I'll show you in a second, by cytokinin. And the type B's are not transcriptionally induced by cytokinin, but are activated by phosphorylation. Okay, now the final difference is once we identified these and looked in the genome, the question is what exactly did they do? And to make a long story short, we basically knocked out all these elements and asked what effect that had on exogenous cytokinin. So on the top there's just seedlings, wild type seedlings treated with cytokinin. The easiest thing to quantify is root elongation, shown on the bottom. If you give higher concentrations of cytokinin, the root growth is inhibited. We quantify that for the type A loss of function mutants. This is a quadruple mutant compared to wild type, grown on increasing concentrations of the cytokinin BA, and we're just measuring root elongation, or actually I guess this time we're measuring lateral roots, which is just another measure of cytokinin response. And you can see a hypersensitivity to cytokinin. So that implies loss of function mutations of these elements increase the sensitivity to cytokinin. So these are negative regulators of the cytokinin pathway. They are very largely functionally overlapping. None of the single mutants really had much of a phenotype at all compared to the higher order mutants. And if you knock out more and more and more of these, you become more and more and more hypersensitive to cytokinin. So these are at the 10,000 foot level, functionally overlapping negative regulators, feedback negative regulators of cytokinin signaling. And then this, the other clade, those type B clades that again had a DNA binding domain, they had the opposite phenotype. So here's the triple loss of function. Here's a wild type plant being sensitive to cytokinin and a triple type B loss of function that shows resistance to cytokinin. So in addition to all the other differences between those two clades, the type A's are negative regulators and the type B's are positive elements in cytokinin signaling. Okay, so again, to make a long story short, work from our lab and other labs have elucidated really the canonical basic cytokinin signaling pathway, which I'll summarize here. The receptors are called the AHKs. They're sensor histidine kinases. They're residing primarily in the endoplasmic reticulum, although there is some debate in the field that perhaps some of the receptors are at the plasma membrane, and that may be functionally important. But minimally, I can say that the vast majority of the receptors are at the endoplasmic reticulum, which, by the way, is the same place where the ethylene receptors are. Cytokinin binds to a conserved domain called the chase domain, which is either in the ER lumen or extracellularly, depending on what you believe. Binding of the cytokinin then initiates the autophosphorylation of that histidine in the histidine kinase domain. The phosphate then is transferred intramolecularly to a fused receiver domain to the AHKs. Phosphate then moves to these phosphoshuttle proteins called the AHPs, which constantly move in and out of the nucleus. In the nucleus, they phosphorylate the type Bs. Again, I should emphasize this is the very same, these are not kinases, it's the same phosphate moving from start to finish. Phosphorylation of the type Bs then induce their ability to bind DNA. They bind to DNA and turn on all the genes that are involved in the response to cytokinin. One set of those genes and the very early response genes are in fact the type A AARs that I showed you before. Those themselves are also phosphorylated by the AHPs, and their role, at least partially, is to negatively regulate the pathway. So they act as, let's say, feedback negative regulators. And we think they also, as I'll tell you in a little bit, probably also interact with other proteins in a phosphodependent manner. And then in addition, there's other players. So this is a multi-gene family in Arabidopsis and in rice, as I'll talk about in a second. There's other more minor players. There's a pseudo-AHPs that negatively regulate the phosphotransferase in the AHPs. There's E3 ligases that result in the turnover of the type Bs, and there's probably other elements as well that we don't know about. Okay, so this is sort of the known canonical pathway for cytokinin signaling. So I want to go in a little more depth today about these type AARs and some thoughts about what they do and how they work. And so it turns out that these are highly regulated, both at the level of transcription. So I kind of showed you this for AR5, but most of them are actually transcriptionally induced by cytokinin, and most of the induction is fairly rapid. So we're looking at 10 minutes. Many of these genes are upregulated in response. So they're regulated at the level of transcription, which we've known. Their transcription is also regulated by many other different things. So, for example, this is actually from rice, but it doesn't matter. This is just again, nobody does this anymore, but it's called nanostrings, where you're measuring the relative expression or transcript level of different proteins. The line here is that it's not induced in the control versus the auxin or ABA. This is twofold, the dotted line, and anything outside that dotted line probably means it's significantly upregulated or downregulated in response to the thing. So the point of this is that if you look at all the type A's, and the type B's are on here as well, some of the type A's are induced, for example, by auxin. Other type A's are induced by ABA. So gets to this question of why a large gene family? Why are there 10? And as we'll see in a little while, there's also there's 11 in rice, and the expansion of these families happen independently in the monocots and dicots. Why? Why are there so many? And one answer to that might well be that different elements or different members of the gene family respond to different outputs. So one might be auxin inducible, one's ABA inducible, and this auxin induction is really important. For example, there's some beautiful work that the auxin induction of a type A plays a role in the formation of a vasculature. They're also induced differentially by exogenous environmental cues. So, for example, high salt strongly induces AR4 and 6 in shoots. Pathogens induce it. Cold induces different elements. So all these different genes are induced by different things, and suggests that one of the reasons this gene family expanded was to provide difference or provide different inputs from or different integrations of different external and internal signals. Okay, so in addition to be regulated transcriptionally, these genes were also regulated post-transcriptionally. This is work from a grad student of mine quite some time ago, but it will be relevant in a second. So basically, this is in the after addition of cyclohexamide, we're looking at the turnover of these proteins in the presence and absence of cytokinin. So the dark line is the absence, and the dotted line is the presence of cytokinin. And you can see that the top three are strongly upregulated or strongly stabilized in response to cytokinin relative to the absence of cytokinin. Others not so much, but certainly a subset are highly stabilized by cytokinin. This stabilization we looked at carefully in the case of AR5 is a result of the phosphorylation. Remember, I told you that the type A's are phosphorylated by the AHPs in response to cytokinin. If we look at, again, here's the wild type in the presence and absence of cytokinin. We're looking at AR5. If you mutate the phosphoacceptor aspartic acid such that it no longer can be phosphorylated to an alanine residue, you completely lose the stabilization in response to cytokinin, which strongly suggests that phosphorylation in response to cytokinin is required for that stabilization. And conversely, if you make a phosphomimic, where you change that aspartic acid to a glutamic acid, which mimics the phosphorylation, you get a stabilization. So these proteins, in addition to being transcriptionally regulated, are post-transcriptionally regulated, at least through the level of phosphorylation. Now, in addition to that level of phosphorylation, we also more recently have looked at the role of autophagy in regulating type A function. And most of you are probably familiar with autophagy. It's basically a cellular recycling system where under conditions of nutrient or carbon stress, the plant breaks down, or the organism, the cells break down various components to reuse the parts, basically, to say it simply. And it involves the uptake of components and the degradation in the vacuole. So we asked the question whether or not type A proteins are also regulated by autophagy. And there was reasons we did this, and part of it was that many of the things involved in cytokinin overlap with what happens in autophagy. And probably the best example is senescence, where senescence involves autophagy, and cytokinin has been shown to regulate senescence. Okay, so one way to do that is with a chemical called conc-A, which basically blocks the autophagic degradation of proteins. And so we looked and asked, if we add conc-A, do we actually stabilize the proteins? And in fact, you do. So this is AR4, one of the type A's that if you remember, go back right quick. This is AR4, which actually was destabilized by cytokinin. It certainly wasn't stabilized. It might, in fact, be destabilized in response to cytokinin. So AR4 is highly stabilized when you add conc-A and inhibit autophagy, suggesting that in the normal course of events, it may be turned over by autophagy. And indeed, if you look at an autophagic mutant, ATG5 lacks autophagy. You see an increase in this, what Western blot, by the way, for AR4 GFP. You can see that when you lose endogenous autophagy, you upregulate type A AR function, in at least this case AR4. Okay, that's all well and good. Does it really go into autophagic vesicles? And the way to do that is to add conc-A and see if you can see it in vesicles, and indeed you do. So this is, again, that AR4 GFP. This is a control. When you add conc-A, you see the appearance of all these small vesicles. Can't show you all the data, but they co-localize with autophagic vesicle markers, and that co-localization disappears in a mutant that doesn't make autophagic vesicles, ATG5. So it tells us that the type A's, at least a subset of the type A's, go into autophagic vesicles, and their turnover is regulated by autophagy, in addition to the regulation by what I showed you previously, which I didn't mention, is 26S proteasome degradation. Now, if you remember last time when I told you about AR5, it seemed that the phosphorylated form was stabilized. Here, it's the opposite. Here, the phosphorylated form is trafficked more frequently to the autophagic vesicles than the wild type, and even which is trafficked more than the D to A version. Again, a phosphomimic and someone that can't get phosphorylated. So here, phosphorylation appears to play the opposite role that it did what I showed you previously, which again, I forgot to mention, but it was a 26S proteasome degradation. So this leads to this sort of complex model where the type A's, again, are induced in response to cytokinin. They're transcriptionally induced, but the unphosphorylated type A's appear to be degraded by the 26S proteasome. In response to phosphorylation, you stabilize a subset of the type AARs. But that phosphorylated type AARs then becomes targeted by degradation by the autophagy. And I didn't show you the data for this, but we found that XO70 is a receptor that targets the type A's to the autophagosomes. So this shows that there's a complex interplay between the regulation of the proteins, both when they're phosphorylated and not phosphorylated, suggesting that the level of type A proteins is absolutely critical to proper plant growth and development. Now, I'm going to come back to the type A's when I get to the end of this, and I'll show you why. But this will sort of answer the other question of exactly how do they negatively regulate cytokinin signaling. So the other thing that we've done relatively recently was we've tried to find additional elements that are involved in cytokinin signaling. And at heart, really, I guess what I am and what I really love is genetics. And so we designed a genetic screen to try to search for such elements. And that screen was to find things that enhance a mutation that is disrupted in critical cytokinin signaling elements, but not disrupted enough to have a phenotypic effect. So we started with the mutant. Again, this is just a re-elongation assay in increasing concentrations of cytokinin. There are 4 functional AHPs. These are the phospho-shuttle proteins in the middle. There's 4 functional AHPs. If you knock out 1 or 2 of those, you see very little effect in this particular dose response assay. However, if you knock out a 3rd one, all of a sudden you become insensitive or partially insensitive to cytokinin. So this is what we would call a sensitized genetic background, where this mutant is decreased in its ability to transduce a cytokinin signaling, but not enough that we can see a phenotype. And the idea would be if we could find a weak mutation that might enhance this phenotype that we might not have found through this screening a wild type plant. So it's screening for an enhancer screen, basically. And we use this concentration here, which we thought was a nice signal to noise ratio in terms of looking at it. So we identified a lot of these. We identified, after rescreening and everything, about 150 independent, what we call EAH mutants, enhancers of AHPs. After we remove the ions, one of the things you find very frequently in these screens are ethylene insensitive mutants, because the reason that the root gets shorter is because cytokinin induces ethylene biosynthesis, which inhibits root elongation. So we found a lot of ions, we got rid of those, and we really focused on things that were not ethylene insensitive. But we got 150 lines. That's a lot of lines, and what do? Typically what a geneticist would do would they pick their 4 or 5 favorite lines and clone them, and figure out who's dominant, who's recessive, and find out how many complementation groups. But we were, I guess, lazy is maybe the best term. And we decided, rather than just pick our few favorite lines, let's take all 150 lines and move them forward and try to identify the causative mutations of all 150 AHP lines. And I don't have time to tell you about this today. I'll be happy to talk afterwards if somebody's interested. But we used a technique that we now dub mutagenomics, where we sequenced all 150 lines and put it through a particular pipeline that helped us to identify the causative mutations in many of those lines, not all of them, but many of them. So the first part of our pipeline, the first thing that comes out are genes that we knew would be involved in the process. And the one that we thought, and in fact turned out to be so, would be the most common mutation is the cytokinin receptor AHK4. Why? Because it's the large gene. The coding region's around 3,000 bases. The whole gene somewhere is around 5 or 6 kb. But also it's the predominant cytokinin receptor in roots, and loss of function mutations in AHK4 give you remarkably long roots compared to the wild type, nearly completely insensitive. So we're not surprised to find lots of alleles of AHK4. And in one sense, we can almost use this as sort of a mini structure function study. And some of the things that we found that were interesting, we actually mutated the aspartic acid residue that is the target of phosphorylation. We found 2 independent missense mutations where we changed that to an asparagine residue. We found lots of mutations, other mutations in the receiver domain. The ones in gray, by the way, are just nonsense or splice site mutations, so not informative in the sense of structure function. The other thing we found a lot of that was a little surprising, we found a lot of mutations here in the early transmembrane domain before the chase domain. The things that we knew that were functional, we knew that the chase domain bound cytokinin, we knew the histidine kinase initiated the phosphotransfer, and we knew the receiver domain accepted a phosphotransfer from the histidine residue in the AHK. So we didn't really find much in the histidine kinase domain, but we found 4, actually 5 mutations that affected this first transmembrane domain, perhaps suggesting that the protein doesn't traffic correctly or in some way, shape, or form cause protein dysfunction. And then the last thing that was interesting is you'll see the 2x here means that we found 3 independent mutations twice. The same exact mutation 2 times, meaning that this screen is remarkably, but the screen is saturated because we're finding the same nucleotide changes in independent lines. We know they're independent lines because we sequenced the genome, and the only mutation that these 2 lines share, they're from independent pools, so one would expect them to be independent, but the only mutation that they shared was the causative mutation. The other 150 mutations floating around the line were not shared in common between those lines. So we found the same hits 3 separate times independently. Okay, so that's not all that great as we expected it, but that's not too surprising. We also found other things that we knew, like the other AHK receptor we found some alleles of. We found a type B AAR. We found OX1, which is known to be ethylene insensitive in roots, and so I would have anticipated that. We found ACS7, which I didn't really expect, but told us that this particular ACC synthase is a target of cytokinin and root elongation, which is different than the target that we found in dark grown seedlings. If anybody's interested, I can tell you about that one as well. So those were the knowns. But then the question is, what were the unknowns? What did Cheney say that one time? Anyway, what were the novel things that we found? So I won't go through all of them, but one of the novel ones that we found that was particularly interesting was an allele of high 5. And this is sort of illustrates to you the utility of the screen. So here's that 2,3 mutant that we started with that we mutagenized. It looks pretty much exactly like wild type. And if you add another AHP loss of function, you get a longer root. Here's a high 5 tDNA by itself. It gives you a weak cytokinin insensitivity, but in the background of the 2,3, which is what these 2 lines, or which is what this line is, you get substantial cytokinin insensitivity, which is exactly what we were screening. We're trying to find things that we probably wouldn't have found this as a mutant, but this would be pretty easy to screen through and find. So that illustrates the utility of the enhancer screen. Okay, so what is high 5? High 5 is a transcription factor that integrates many different signaling inputs. It certainly was first identified as a role in light responsive gene expression and morphogenesis, but it's been linked to many other plant hormones. So downstream of high 5, ABA, auxin, GA, brassinosteroid, and ethylene have all been linked downstream. Upstream of high 5, cytokinin via COPS, but ethylene and GA and strigolactones have all been linked. So it's an integrator of many different hormone signals. So the question is, how much of a role did it really play in cytokinin signaling? So we did 2 things. We did chip analysis, where we tried to ask the genes, what are the genes that are targeted by high 5 in the genome, and how do they compare to the genes that were targeted by type Bs? And we found a remarkable overlap of almost 60% of the genes that were targeted by high 5 were also targeted by type BARs. What that says is that they have a large overlap in their target genes that these 2 transcription factors hit. Likewise, of course, if that's true, one would expect that there would be changes in gene expression, or high 5 would affect gene expression. And here what we're looking at is genes that are induced by cytokinin in a wild type plant and genes that are induced by cytokinin in high 5. You can see, again, many of the genes overlap, but there's 252 genes that are no longer induced by cytokinin in high 5, and then another 500 or so genes that are now transcriptionally induced by cytokinin in the high 5 mutant that aren't transcriptionally induced in the wild type. So it seems like high 5 plays an important role in mediating the transcriptional response to cytokinin. And I want to go from this to sort of a bigger idea without a whole lot of data, but I'll just give you our hypothesis and where we're going with this. So one of the questions I started with is, how does one molecule do so many different things? And then secondly, how do the type A's negatively regulate cytokinin signaling? So one of the answers in mammalian cells and other systems is how a single molecule can do so many different things is the presence of partner transcription factors. It defines what is called context-dependent signaling, that there's a primary target of a signaling pathway. In our case, it's the type B ARs. And the suites of genes that are regulated by the type B ARs is determined by their interaction with a series of other partnered transcription factors. And all those transcription factors shown on the left have been identified as factors that directly interact with type B ARs by our lab and other labs as well. The one I just showed you is high 5, but we identified CRFs and BPCs and IN3s. Deltas have been shown to interact with type Bs. All those elements on the left, or transcription factors, have been shown to interact with type BAR. So our hypothesis is that it's the combination of cytokinin signaling with the partner transcription factors that determines the precise output of the cytokinin signaling pathway in a particular cell or tissue type, and that it's the combination of these two that then determine the function of cytokinin in that particular cell type. So that is one hypothesis that we're currently addressing for how cytokinin can do so many other things. Other possibilities include the fact that there's multiple type Bs and multiple AHKs, and then the role of chromatin is probably important as well, but this we think plays a role as well. But the other thing this got us to is this idea of, again, we knew early on that the type A's played a role as a negative regulator of cytokinin signaling, but we didn't, and we still don't really know the mechanism. So one simple idea was that the type A's just heterodimerize with the type B's, which is how the OXIAs and the ARFs work. That would be a simple idea, and that this functionally binds DNA and this doesn't, and the heterodimer wouldn't bind. That doesn't seem to be the case. We and many other people have looked for interaction of the type A's and the type B's and have not found any. A second possibility that they just compete for phosphoflow from the AHPs, but we've also ruled that out as a sole mechanism because we know that the phosphomimics of the type A's, which can't be phosphorylated, still are functional. So then what then? How do they work? And one idea how they work, and I will stress this is at this point mostly a hypothesis, is that they compete for binding to these partner transcription factors. What do we know so far? We know that the type B's interact with the transcription factors shown here. And we know that the type A's also, we did a large screen, exhaustive 2 hybrid screen with a set of type A's. And Thomas Schmuling had done a screen, another cytokinin researcher in Germany, did a screen for the type BARs. And we found a series of overlapping transcription factors that interacted with both. And so we're testing the hypothesis now that the way that the type A's act is by competing for interaction with these partner transcription factors. But again, I want to stress it's just a hypothesis. We don't actually have the answers yet. Okay. So this is sort of a summary of the work with type A's. So I first told you that the type A's are redundant, partially redundant negative regulators, and they act as feedback inhibitors for the cytokinin pathway. They're regulated at multiple levels: transcription, protein stability, and by phosphorylation. They're also regulated by both exogenous and endogenous signals. They act to probably integrate multiple different signals to modulate the cytokinin sensitivity of a particular cell or a particular tissue. And then finally, they may act by competing with type B's for partner transcription factors. So that's sort of where we're at with the type A's. And how long do I have you again? I've been paying attention.
Well, we still have 10 minutes.
Just 10 minutes?
Yeah.
Ooh, that's not a lot. Okay. Well, I'm going to tell you quickly about our work on rice then. So almost all the functions of cytokinin were delineated in the dicot Arabidopsis, right? And we were interested in moving into a monocot, and we chose rice for a couple of reasons. One reason is that rice is, I wrote on here. Anyway, rice accounts for about 23% of all the calories consumed by humans on Earth. The amount of rice made or harvested worldwide has been increasing in the last 60 years or so, but it is beginning, at least in China, to plateau out. So there's that combined with global climate change and increasing climate means that we need to make more rice to feed the world. And so I won't go through this, but climate change has a very strong negative impact on rice yield. So there needs to be ways to increase rice yield to keep up with the increasing human population. So rice in particular plays an important role in yield in cereals in general. There's some beautiful work from Matsuoka's lab in Nagoya, where he showed that there's 2 main varieties of rice grown in the world, Japonica and Indica, the Indica being a higher yielding variety. They crossed those 2 together. They identified a single QTL that accounted for about 80% of the yield difference. That QTL was a loss of function of a gene called cytokinin oxidase, which is a gene that degrades cytokinin and results in the high yielding variety having a higher level of cytokinin during the early panicle development, presumably increasing cell division and the formation of spikelets and yield. So we think cytokinin can play an important role if manipulated appropriately to increase both rice yield and perhaps other cereals as well. So this is a cytokinin signaling pathway in rice. It looks just like Arabidopsis, just slightly different names. So our approach was to knock out, using CRISPRs, the different cytokinin elements. So we've knocked out all 4 receptors, both AHPs, the 3 PHPs, and all 10 or 13 type A's, and as well as our collaborator has knocked out the type Bs and asked what happens to rice growth and development. So I'll tell you first the work we've done with AHKs. So if you look at the phylogeny of the Arabidopsis and rice AHKs, they interdigitate nicely. And you can see clear orthologous relationships, suggesting that the progenitor of monocots and dicots probably had 3 receptor genes. If you knock these genes out, you see an increasing severity of phenotypes from the singles. We have all of them, but I don't have pictures for all of them, but it holds up with others. From the singles to the higher order mutants, culminating with the quadruple mutant, which basically doesn't make a shoot at all and makes at best an abbreviated root. And I'll go through this in turn. Shoot, same thing, increase the loss of function AHKs, you end up with a tiny little shoot. If you knock them all out, you end up with no shoot, but you knock out 3 of the 5 or 3 of the 4, you get an abbreviated shoot. I'm gonna run out of time, so I'm gonna go through this pretty quick. They flower much later than their wild type counterparts if you knock enough of them out. And there wasn't a whole lot of indication that cytokinin regulated flowering time in Arabidopsis, but it plays an important role in rice, it seems like. Trichome formation is defective in the higher order mutants. These are the rice trichomes. This is the surface of a leaf. Those are the trichomes in a wild type, but they're nearly absent in the higher order mutants, and we quantified that. The number of tillers is substantially reduced to the higher order mutant, important agronomic trait relative to the wild type. Again, this is just tillers over time. Wild type, the lower order mutants don't have a strong phenotype, but some of the combinations have a strong phenotype, particularly the 3, 4, 5. The angle of the tillers seems to be different. This is a wild type plant. You can see that the tillers are coming out at maybe whatever that angle is, but you can see that 3, 4, 5 is a much more upright plant. And again, tiller angle is a really important trait for rice in the field. But so some of those things are very different than what we found in Arabidopsis. Some are the same, but the most dramatic difference comes here with the quadruple loss of receptor function. So here, this is a triple loss of function where 3 of the 4 receptors are knocked out, 2, 4, 6. This is a quadruple mutant. And these plants are actually 2 weeks older than these plants. This is about as big as they ever get. Sometimes they never make a root or shoot. Sometimes they make a little tiny abbreviated root, but they never make a shoot at all. Now, if this is the plant that I started with, I would have said, of course, they're not going to make a shoot. I told you way back when in the 50s, Skoog and Miller showed that cytokinin is essential to form shoots. And this then would be the culmination of that work. However, if you look at, so this is just a close-up of it, it never makes a shoot, very unhappy little guy. If you look at Arabidopsis, and we've done that, we've actually made triple null mutants where we know that they're nulls. They make absolutely no functional protein. Those plants still make a shoot. Now, albeit the shoot is smaller, they still have trichomes, they still make a shoot, and they make a reduced root, but they do have a shoot. It still makes several sets of leaves. So it's a very different situation between rice and Arabidopsis. And if you want to extrapolate that to monocots and dicots, in the case of rice, cytokinin is essential for shoot formation. In the case of Arabidopsis, it's not. And one of the reasons that it's probably essential in rice is that if you look at the size of, in this case, the inflorescence meristem in a double mutant, it's greatly reduced in size. And what we're currently doing is we're trying to section through early embryo development and see if that shoot apical meristem disappears in the HK quadruple mutant. Okay, I'm going to run out of time, so I want to get to the type A's, but that affects the formation of the inflorescence. As you knock out the type A's, the inflorescence gets really smaller. If you knock them all out, you get no shoot. Affects flower development, stigmatic brushes. Again, this is not a phenotype you see in Arabidopsis. Even the triple receptor mutants, when they do flower, you don't really see effects on floral formation. Affects seed size, which you do see in Arabidopsis, but the single loss of function mutants for the HKs have bigger seeds than the wild type. And the roots are strikingly affected. Partially the result of meristem size. Okay, the last couple minutes, let me tell you about, so that's the AHK. So the AHKs, again, as in Arabidopsis, seem to be fairly pleiotropic. They affect root development, seedling development, shoot development, inflorescence, and floral development. So the thing we've done most recently is we've been started to work on the type AARs, which are a large gene family, and we split them into 4 independent clades and knocked out each clade in turn. One of the things I want you to get from this is very different from the phylogeny I showed you with the AHKs. The AHKs from rice and Arabidopsis more or less interdigitated. Here, the Arabidopsis and rice type A's clade completely separately, suggesting that these two gene families expanded independently in monocots and dicots or after the monocot and dicot split, which might be the fact that these things are induced by different environmental and endogenous signals I talked about before. So what are the phenotypes of the type A's? This is really just starting, but one of the interesting phenotypes we see very clearly in this particular clade here, this quintuple clade right here, when we knock out all 5 of these genes, we see the twinning of the embryos. We see about 70% of the embryos are either twins or even sometimes 3. We see other aberrations in the shoot as well, but the most common thing we see is twinning. And we're trying to figure out why that is. Again, in contrast to the AHKs, we see more trichomes relative to the wild type. Quantified that here. So there's more trichomes, the opposite phenotype of what you'd expect from the AHKs, which HKs are insensitive. These are hypersensitive, so you'd expect more trichomes. If you look at the inflorescence, one of the slides I showed you when I started the rice discussion was that loss of function of a cytokinin oxidase, which degrades cytokinin and had a bigger panicle. So one anticipation might be if you knock out the right combination of type A's, you may increase panicle size. And in this particular quintuple, indeed you do. You have more spikelets and you have more secondary branches than you do in the wild type. So again, we're in early days, but I think different combinations might even give us a more dramatic effect, maybe something approaching what we see with the indica japonica varieties. Okay, so just to summarize our rice work, and sorry I went through that pretty quick, but the basic idea is that what we found is that the AHKs redundantly regulate cytokinin sensitivity in rice, just like they do in Arabidopsis. And the 4 AHKs play distinct but overlapping roles in many different aspects of development. Some of the roles that we see in rice are similar to what's in Arabidopsis, for example, relongation, but some are very more prominent in rice. So there is sort of some indication that trichomes might be affected by cytokinin in Arabidopsis, but they're absolutely affected in rice. And some are really pretty much unique. First of all, the complete lack of a shoot in the quadruple, and some other phenotypes that we see have no comparators to the Arabidopsis. And most interestingly, as I said, knocking out all 4 of them results in a completely shootless phenotype, which is very different than what we see in Arabidopsis. And finally, the type A's also negatively regulate cytokinin signaling in rice, just like they do in Arabidopsis. And they also affect multiple aspects of development in a functionally overlapping way. And some of the things that we've looked at, I've shown you, and some I haven't shown you, but it includes tiller number, chlorophyll content, leaf development, panicle architecture, trichome density, and seed size, as well as other phenotypes as well. So we're really kind of early days in quantifying the developmental effects of these. So really, that's where we're at now. What we really are going to try to do is we're going to now go back into, where am I here? Into our current work. If I get to this, we're going to go back to these inflorescences, both the vegetative and the inflorescence meristem, and do like everybody else on Earth, the single cell, to try to find out, first of all, in a rice panicle, defining the kinds of cell types and their developmental trajectories, but then also try to figure out how cytokinin alters that. And then we're going to continue to characterize these mutants and identify the different phenotypes. So that's where I'll end. And I'll just tell you the folks that did the work. Eva is been in the lab for about a year or so. She's a great graduate student. Emily actually was an undergraduate in my lab, went to Michigan State and has come back as a postdoc. And then these are some undergraduates that have worked in the lab, and some of the folks that started the rice work in my lab include Christian and Kevin had done that. I'm sorry, Asha. And then these are our collaborators. We collaborated with Eric for a long time on cytokinin signaling. Josh Strabel, who was in Mike Scanlon lab, is doing the shoot apical meristem single cell with us, and some of the other folks that have been collaborating with us are shown up here as well. So I'll stop there and be happy to take any questions you might have. Yeah, Mike.
So the type A mutants in rice that which went to, so they also have a larger meristem? Is the polypeptide sequence?
I don't know yet, to be honest with you. We literally just started doing those sections about 2 weeks ago. I don't know the answer. the hairy sheath mutant in maize, which is a gain of function mutant in cytokinin receptor. According to Josh, anyway, it's not published, but according to Josh, that also showed twinning in maize.
Because it's a single mutation of type A in maize.
Yeah, ABFIL1, yeah. Yeah, but I don't think it showed twinning, did it? The ABFIL1?
The main culprit is the large marrow.
Yeah. So we're looking at the barossome size right now, but I assume that they do, but I don't know the answer to that yet. But yeah, it's a good question. Yeah, just.
So I really like this transcription factor model with the response regulators interacting with this group of TFs because it kind of helps understand how everything's doing everything. But so I'm curious, what how complicated do you think that is in terms of, are these all 2 molecule reactions, or do you think they're more complex than that? And I guess the rest of the question is, I assume those were things that you showed you had interaction.
Yeah.
You probably don't need to have direct physical interactions
Of course.
to still work.
Yeah.
And there's probably a lot of other TFs you could infer should fit into that group. So I'm just kind of wondering what are you?
Yeah, I mean, you're thinking exactly the same way we're thinking in exactly the same direction. I'm guessing it's going to be remarkably complicated. At a single cell level, maybe not so much. Maybe at a single cell level, there'll be a suite of partner transcription factors that interact with the type Bs under particular conditions. And as you say, it could be that they directly interact with the type Bs. It could be that they're part of a large complex, or it could be that they independently hit independent cis-acting elements and just co-regulate in that way. And that's the way it works in animal cells. So I mean, we're very early days, but those are the exact. So one of the first things I want to do is I want to take a reporter line with particular targets for type Bs, and then just dial up and down type A function and see if this hypothesis is right. Because at this point, it's just a hypothesis, really. But it's got to be something like that, because the specificity, it does so many different things. It has to be partner transcription factors, and I can't find a particular other mechanism by which type A's negatively regulate the pathway. I think it would nicely solve both those problems, or at least address both those questions. But it's early days, and my guess is it's going to be both what everything you said was probably going to be true. It's probably simple heterodimers with the type Bs and the partners. It's probably going to be large protein complexes, and I also think there's probably going to be independent targeting of cis-acting elements as well. But that's where we're heading. Sorry, next branch. Yeah.
I also have a question concerning the spatial regulation and different function of cytokinin in the plant. Because I mean, you did talk about that transcription factor combination between type A and type B. I wonder, because you also showed those 5 different mutants in the AHKs.
Yeah.
Are there, or how many homologs do you usually have of AHKs within one genome, basically? And do you see if there's different mutations in those homologs, and there's basically different AHKs with different alleles expressed in different?
Are you talking about in different plant species? Is that what you mean? If I look across the phylogeny of plants, do you see different AHKs in different places? Is that what you're asking?
More within one genome. Let's say you have, I don't know, 10 copies of or 10 different AHKs that have that have mutations and are thus expressed in different tissue types, and then have different downstream effects. That's how you have the different.
Yeah, I mean, that's certainly part of understanding how one signal does different things, is that you have multiple receptors and multiple AHPs and multiple type Bs, right? So the combinatorial diversity of in the case of Arabidopsis, it's 3 AHKs, 4 functional AHPs, and remember, right, 11 type Bs, and you do the math, that's a pretty large number of different combinatorial outputs. I think that's part of it. I do think that's part of what imparts specificity, but we also know that there's a huge amount of functional overlap. So for the type A's, if we knock out single type A's, we see almost no phenotype, and there's actually oddly enough, relatively little information gained by patterns of expression and who's functionally overlapping with who. And I would have anticipated there would have been, but it doesn't seem to be. And that's true for the IPTs as well, the biosynthetic enzymes. Part of that answer might be that if you knock out a suite of type A's, you get compensatory upregulation of some other type A's. You know what I mean? So I'm not quite sure I'm answering your question, but certainly we do think that tissue specific expression of different components within the pathway contributes to specificity of function. I think that's certainly true, but that's only a small part of the answer. It has to be something else as well. I'm not sure that answered your question or not, but we can chat later if you want, but yeah. Any other questions?
So it's a question online.
Oh yeah, how do we do that?
City.
I don't know. I'm still stuck in the first slide here. I don't know why that's different from that. Yeah, I don't know what's going on there, Mitch.
Well, I don't know how to do that. Anyone you log into that Zoom, right? Oh, no? Okay.
Don't worry about it, I guess.
Oh, there it is.
Oh, okay.
What do I do now?
Yeah. On the chat. Yeah, click on the chat.
Ah, there we go. All right. Yes. I say yeah. Yeah. So actually for several of the type A's, we haven't actually done this for the type B's a lot. The interaction definitely depends on phosphorylation. If we look at the phosphomimic versus the wild type, we see much stronger interaction with the phosphomimic than we do for the wild type, but that's only true for a subset of the transcription factors. And we haven't actually explored that much with the type B's, but yeah, that's a great question. And that's absolutely true that there is some of that. Speaker audio is not working. Oops, too late now. Anyways, that's a great question. Yeah, we actually definitely see phosphodependent interactions.
Okay, any other questions?
Question that bugged me for a while. Well, first of all, thank you for a wonderful talk. I'm sort of struck with the fact that the receptors for cytokinin and ethylene, they are located in the ER.
Yeah.
And it's always bugged me, why ER? Like,
My grandmother used to say that's the way God made it. For the case of ethylene, it probably is fine, right? Because ethylene is freely permeable to the membrane, and actually ethylene binds within the membrane to the receptor. So it's not surprising that it would be an internal membrane for ethylene. The cytokinin ones, I don't honestly have a good answer for you. The first papers that came out for cytokinin showed that it was in the plasma membrane, which is where everybody expected it to be. But if you look at the high affinity cytokinin, but the best work on this has been done by Thomas Schmuling, where he localized both GFP fusions, but he also looked at, in a wild type plant, high affinity cytokinin binding and saturable high affinity cytokinin binding, and almost all associated with the ER. So I think it's clear that's where it is. I think there might be a small, I mean, it's hard to argue that a small fraction of it doesn't go to the plasma membrane and that part's functional. I can't rule that out, but certainly the majority of the AHKs are at the ER. Why that should be, I honestly don't have a good answer for you. It bugs me too, but I don't know why. I mean, maybe because there's crosstalk with the ethylene receptors, and that's where the ethylene receptors are. That might be the most simple answer, but I don't really know if that's true or not. But I don't know. Yeah.
In that realm, I'm wondering how the cytokinin gets into this now.
Yeah.
Yeah,
How does it get in, and how does it get into the lumen of the ER? Because it has to bind in the lumen of the ER. So there's been multiple potential transporters identified, like the PUP transporters, which are purine transporters, have been shown to transport cytokinin. Hitoshi Sakakibara has shown some really nice work about some transporters that export cytokinin into the xylem stream. But as far as I know, we don't know the cytokinin transporter that brings it certainly into the lumen of the ER. And that was one of the things I anticipated maybe finding in our screen, that maybe we would have found some transporters. Also, what's his name? Anyway, so there's a bunch of papers out there about different potential cytokinin receptors. I don't think any of them, as far as I know, are in the ER. And I don't know that there's a smoking gun to prove that any of them actually bring it into the plant cell at the moment. But there's a bunch of possibilities. But yeah, it's a great question. I'm one that bothers me. Both of those questions bother me and keep me up at night. So, yeah.
I'm not sure how the experiments were done in looking at the plasma membrane and the ER.
Yeah.
But was the fact that there's so much more ER membrane than plasma membrane taken into consideration when saying?
Well, I didn't do them, but the two experiments that were done were they localized fluorescent proteins, and the fluorescent protein is effectively 99% of it localized to the ER, and effectively almost none of it was at the plasma membrane. I mean, it's really hard to say that none of it's there, but effectively they saw almost none. For the high affinity binding, there was essentially no saturable high affinity binding that they did sucrose gradients, and they saw no high affinity saturable binding that associated with the plasma membrane. And they said essentially all of it associated with the ER by biochemical fractionation of the membranes.
Okay, great. Many thanks for your.
This has been a production of Cornell University, on the web at cornell.edu.